All right, in this chapter, students, we're going to talk about how are genetic diseases treated. Technology is moving at a pretty rapid pace, and we now have the ability to treat some genetic diseases, whereas we didn't have those abilities in the past. Well, how do we get DNA obtained for genetic testing to test for if individuals have a certain genetic disorder or not? Well, we can do this in adults using a blood test. We can use amniocentesis and chorionic villus sampling to get genetic material from a fetus, and both of those processes are shown there. How does a karyotype, and this is a karyotype here. A karyotype is the image of a person's chromosomes. You would go into a facility, they would take a cheek swab, they would multiply your DNA using PCR maybe, and then they would fluoresce certain parts of your chromosomes and they would highlight them, and then they would kind of pair them up, and you can see how this pair, you have a little pink part there and a little pink part there, so they kind of match up. And they can look at this karyotype here to see if there are any different structures to the chromosome. Maybe a piece of a chromosome is missing. If you have an extra 21st chromosome, that would mean you have a condition called Down syndrome. And in this case, the person is X and Y, and that would make them male. It is possible to clone a gene into bacteria too, and let the bacteria make multiple copies of that DNA. So you would have a human cell here, and this human cell has an insulin gene on it there. And on that insulin gene, we could remove it with some procedures, take a bacterial host cell, take this little plasmid, which is an extra piece of DNA that bacteria carry, stick that insulin gene into the plasmid. This bacteria will then multiply, and when it multiplies, it will begin to produce insulin. And these bacteria will also make cloned insulin genes that we can use for study as well. It's possible to know certain diseases may be associated with the inheritance of a genetic marker. It's a variation in the pattern of DNA. Sometimes these variations occur outside of genes and sometimes within genes. We use special enzymes called restriction enzymes that break the molecule down at particular base sequences. DNA sequencing determines the order of nucleotides, and by doing that, and you can actually get online and look up human genome, and you can actually see the sequences that scientists have used that they collected in the past. So what is gene therapy? Gene therapy involves the insertion of genetic material into human cells for the purpose of treating a genetic disorder. Well, the goal here is to take a gene, like the gene for sickle cell anemia, and replace it with a gene that does not have that nucleotide sequence that causes hemoglobin to sickle cell. So you would take a gene that causes sickle cell, and then you would replace it with a gene that does not do that. We can see here, the process of that. Remove bone marrow stem cells, use retroviruses to bring the normal gene into the bone marrow stem cells. The virus itself will then go in and cut out the bad gene and replace it with the good gene, and then return those genetically engineered cells to the patient into their bone marrow and hope that they produce proper red blood cells. Well, Scientists have been working on methods to cure sickle cell disease by correcting the defective gene outside of the body and then reinserting it into the cells. And so one of the problems with that is, how do you get to every cell in the bone marrow? It's hard to do, and you have to use some type of virus typically. And what we don't want to have is the virus mutating in some way and then causing harm to the patient. Doctors say the first rule is do no harm. And so we can do genome editing, and genome editing is pretty exciting because of this new thing called CRISPR. It's based on an enzyme called Cas9, and Cas9 is capable of identifying specific sequences of nucleotides, breaking both of the strands, and then inactivating the virus. And what we can do is we can take this is the guide RNA here. So this would be what we want to have placed in our patient's DNA. This is the target gene that we're looking for. And what this enzyme Cas9 does is it will bind to this DNA. So it will bind here, and it will cut out this target piece. And we have two options. You can either leave the cut and just have this deletion in the target zone, or you can have donor DNA that this Cas9 carries and have it replaced into where you cut. And so now you have this point mutation gone out of the host DNA. Because we know the gene for sickle cell and we know that it's the result of a single mutation, we can do genome editing for it. We've been able to use CRISPR to repair up to 40% of the cells, which in turn reduce the amount of sickle cell red blood cells in the patient's blood. And they're still working on ways to get this number, 40%, up. This question here says choose the correct statements. There is more than one correct answer. And that's all for this chapter. It was a pretty short chapter, but I hope it was exciting.